I am in charge of testing the water on Grand Lake and also doing special projects with our university partners and also our state partners. During the summertime, we're extremely busy this time of year testing Grand Lake, Lake Hudson, and W.R. Holloway pumpback storage facility. We test from Pensacola Dam all the way up to Twin Bridges, all the major coves and creeks. So we test Drowning Creek, Duck Creek, Horse Creek, and also we get call-outs on special situations such as if anyone calls in for blue-green algae they'd like us to investigate, any potential things like that, or even bacteria or some health stuff that happens at public beaches. I currently have 3 interns working underneath me and also a new employee named Richard Zamor, actually Dr. Richard Zamor, who specializes also in limnology and water quality like I do. He brings some different skills, unique skills to the GRDA, as that he is an expert in DNA. In a typical water sample, we're out looking for nutrients in particular. Just like a field of soybeans, the water has phytoplankton, or algae as we refer to them as. These algae require nutrients to grow, so we look for dissolved phosphorus, dissolved nitrogen, total phosphorus, total nitrogen, and that will tell us and give us an idea of how much fertilizer is there, and that will give us an idea of what's the potential for the algae to continue to grow, which can signal, for example, an algae bloom of any sort, whether it's BGA or if it's just a regular run-of-your-mill algae bloom, which occurs every year on Grand Lake. We also test for chlorophyll, water temperature. The chlorophyll will tell us how much algae is in the water. We also will test for bacteria. We test lake-wide from Twin Bridges all the way to Pensacola Dam. Other special projects we have, we're working with the Water Resources Board to do a study on the pump storage facility, looking at some interesting oxygen anomalies that occur extremely deep down in the water.
My name is Rich Zamor. I'm the newest addition to the Ecosystems and Education Lab here. I just started in June, so it's been a pretty wild ride. My background is a limnologist, and so I study the lake, and I'm also really interested in the ecology of the smaller organisms in it, like algae and bacteria. So those are sort of my specialties, but I also have just a general background knowledge of how, say, nutrients in the lake work and other sort of forces like that. For nutrients, we use the Latchet system over there, and so that can tell us different levels of nitrogen, phosphorus, and ammonia, and so those are really important when thinking about when algae might bloom and how energy moves around within the lake between different organisms. We also have the ICP. The ICP lets us look for trace metal contaminants, so from like the Tar Creek Mining District, things like that, and so we can determine if they're affecting, maybe negatively or affecting the organisms in the lake. We also have a trilogy, and so what a trilogy does, it allows us to read the amount of chlorophyll that's in the lake, and so chlorophyll is another way of looking at algae and how much algae standing stock is there. It can also tell us about the productivity of the lake. How quickly or how well are the nutrients turning into new plants, for example, so if you think of algae as tiny plants. We also have just our plate reader system for reading bacteria, and it's a pretty simple system, but pretty effective. We also have the flow cam. The flow cam is really exciting, and so it's part camera and part flow cytometer. And so what flow cytometer means is that water actually flows through the instrument, and it can measure different sorts of cells. It can look at how big they are, and then take pictures of them while it's going. So this allows us to take pictures, or as we're taking pictures of the algae, we can also count them. And so during, say, a BGA bloom, for example, we could quickly count the amount of BGA that's out there so that we can make decisions on that. Also, if it's there at low levels, we might be able to see it quicker than we could, say, using a microscope. This lab is really well equipped to do sort of the initial steps into examining all your limnological parameters. I mean, the equipment in here is second to none that we have as far as analyzing nutrients and trace metals. We're also thinking about incorporating a genetic component, so using genetics to monitor the amount of algae that's out there or the amount of bacteria, and then not only that, being able to determine where that bacteria might be coming from. For example, like a non-point source, say like chicken farms, or say a point source like leaking sewage or something like that from a septic system, or like Canada geese that have taken over a beach. We're working on being able to implement that into our routine monitoring as well. That stuff is on the cutting edge as far as methods being developed for it. It's got precision. So compared to say a microscope, the precision is 100 to 200 times better, because you're actually counting things using their genes instead of using, your naked eyeball looking at what's in a water sample. And so so your limits of detection go much lower. So you could find, say, 1 cell in 1 liter of water, for example. So if you take you think about that 20-ounce Coke or that 1 liter Coke, it's 1 algae cell in there, and you could find it. So and that can be really important when you're starting to look at as nutrients change or as chemicals change, how does our algae community change? So does BGA start up early, when we see some nutrient spike or some chemical change like that, or can we associate things that are like chlorine and different, like chlorine is used to purify water for human drinking water. How does that associate with, say, the bacteria we're seeing? It could be an indicator that we should be, looking for, E. coli from a human source, something like that. So we could really put those two things together and really start making that could really aid in management decisions later on.
The vision for the future for the Water Quality Lab is to gain more knowledge about the projects and the waters that we oversee, to become the experts when it comes to water quality or any sort of environmental issues on Grand Lake. With the tools that we have here, we are just starting to scratch the surface of what we are capable of doing and what we are capable of knowing. This will advocate for our water quality and also the conservation of the waters of the Grand Rivers. With this, we will also be able to make better management decisions regarding shoreline management plan, and also it will give us I think a stronger say as to what can go, as to the future of conservation efforts within our watershed.